Welcome to Unit 6, Lecture 4. This is the last lecture for Part 1. Okay, Part 1 of Unit 6. Remember, Unit 6 is going to encompass 3 related topics. Okay, so it's going to be kind of long. So this is the end of the first part. This is talking about meiosis. So meiosis is a little bit different than mitosis. Okay, so it's going to be very, very important for you to be able to make sure you have your vocabulary clear so you can keep these two things separate. Learning objectives. Identify how many sets of genes are found in most adult organisms. Describe the events that occur during each phase of meiosis. Identify the difference between meiosis and mitosis. Explain how 2 alleles from different genes can be inherited together. Mitosis. Prophase, metaphase, anaphase, telophase. Okay, remember PMAT. All right, so we've got getting ready, nuclear envelope breaking down, lining up in the middle, spirit fingers pulling apart, and then nuclear envelopes reforming, and then with cytokinesis, cell splits. Same basic stuff happens in meiosis. The only problem is it happens twice. Okay, so we get meiosis 1, okay, meiosis 1, and then we get meiosis 2. So the same thing happens twice. So we get prophase, metaphase, anaphase, telophase, and cytokinesis, 1. Prophase, metaphase, anaphase, telophase, and cytokinesis, 2. Okay? The same basic things happen during meiosis that happen during mitosis. Again, I'm going to go through these pretty quick because we're going to actually look at them more in depth in class. All right, so I don't want to spend a whole lot of time talking about stuff. Okay, so same basic things happen. The biggest difference is that in prophase 1, you see this thing called crossing over, which I'm going to talk about in just a second on the next slide. But try not to get so worked up thinking these are 2 different processes. They're very similar. Okay, it's just that mitosis times 2. So here is where I'm going to spend a little bit extra time in prophase 1. Okay, so this is prophase 1. The thing that's different here is so we have this thing here where we've got the big A and the little a, okay, our homologous chromosomes, with their copies, their sister chromatids. This is called a tetrad because there's 4 if you play Tetris. So we've got 1, 2, 3, 4 chromosomes. 1 and 2 are identical. 2 and 3 are, or 3 and 4 are identical. Okay, we've then rubber banded them together at the centromere. During prophase 1, they can switch pieces. Okay, see here, they can actually switch pieces. This is why you might have some things that are very similar to one parent and not the other. Okay, but you might have a little piece that's just like a parent. Now, if you get your DNA done, if you get to do any of the DNA tests, you can see chromosomes and you might be able to see where these differences are. It's pretty cool. Okay, so if we were to look at the red chromosomes, all right, we could say A, B, C, D, the centromere. Okay. The yellow chromosomes we could say is A, B, C, D. All right, so all uppercase, all lowercase. What happens during crossing over is we end up switching the pieces here. So we get one chromosome that's A, capital B, lowercase, and then lowercase c, lowercase d, lowercase a, b. Okay, so I know this drawing is kind of all over the place right now. All right, but what happens is we've actually cut the chromosomes and swapped those pieces together. So you can see here, they now have 2 different spots. So this is a way to increase genetic variation. Okay. Metaphase 1, things still line up on the center. All right. Anaphase 1, things, chromosomes still moving away from the center. Okay, so the thing here is we're pulling sister chromatids apart. Telophase 1 in cytokinesis, nuclear envelope reforms, and we divide the cell. Meiosis 2 starts with these cells, okay, and does it all again. Line up in the center. Now, instead, we're pulling apart the sister chromatids. Okay, so the end product of meiosis is 4 daughter cells that have half the normal chromosome number and they're different combinations. Okay, they're different combinations. That's the important part. All right. So here's a picture that summarizes meiosis. Okay, you see. So, like I said, we are going to talk more about these. I know this is a little confusing, so we'll go over it. All right. See you in class. Have a great day.